Things, of course, next week is what? Midterm, yes. And happy President's Day to everyone as well. As usual, there will be no new material on Friday. That will just be a review, but I will have a couple of quicker questions. So you can come for the first 5 minutes and then take off. If you need to figure studying is better done elsewhere rather than here, I will be posting that lecture online as well, at least the review parts thereof. At this point, any questions about the next exam? It's going to be 50 multiple choice questions. Bring a Scantron. Not per se on material that was covered on the last midterm. Should be just the lectures since that last midterm. Any questions at this point about that? No. Okay. Then let's move on and start our clicker questions for today. Polyamines, like spermine, are common in bacterial cells because DNA is an efficient way to encode genetic material. Bacterial DNA is positively supercoiled. Bacterial DNA is negatively supercoiled. DNA is negatively charged. They have small genomes. Thirty seconds. Ten. So what do we think? That the reason that polyamines like spermine are common in bacterial cells is because DNA is negatively charged. Yes, because you have to be packaging it. The idea here is that DNA is actually a pretty inefficient way of encoding your genetic material. And so therefore you have to compact your DNA. One of the big problems with compacting DNA is that it's got negative charges on its backbone. So one of the ways to deal with that is to be adding a uniform or relatively uniform positive charge, and spermine and spermidine are the two most common ways that you do that. Bacterial DNA in general is negatively supercoiled. Yes, that's true. One of the things actually with negative supercoiling is that depending on your twist and writhe, usually it means you're untwisted a little bit, which actually is going to be pulling the strands apart to some extent, which, if anything, is going to be positive, or not positive, going to be selected for because those two backbones really want to be pushing it themselves apart as well. So our next clicker question is most exon duplication in the evolution of the human genome probably took place by unequal crossing over, homologous recombination, recombination between transposons, A and B, or A, B, and C. Thirty seconds. 10. So now we seem to have much more of a difference of opinion on this one. Okay, a couple things about this question. So most exon duplication, so we're really thinking about the relatively small pieces of DNA which are being duplicated here. Most of that duplication is going to be happening through what? Okay, we all can't decide. Unequal crossing over, homologous recombination between transposons, or some combination thereof. Clearly, to get duplications, crossing over can't be equal crossing over. So it would have to be unequal crossing over. So I think A sounds reasonable. How do you get crossing over to take place in the first place? Homologous recombination. How do you have homologous recombination? What do you have to have? You have to have homologous sequences, or basically identical sequences. What are transposons? Identical sequences. So the answer is A, B, and C here. And we'll be talking a lot more about transposons for the rest of lecture today. So our third clicker question is probably more of a genetics question than a molecular biology question. That there's more genetic divergence in introns and exons means that there is purifying selection in introns, purifying selection in exons, no selection, no molecular clock, lots of speciation. Thirty seconds. Ten. Okay, I agree with most of you on this one, so I like that. The key here really has to do, there are a couple of different things here. One is purifying selection. What's purifying selection doing? Means that things that don't work as well get purged or purified from your environment. So if you have these mutations that are not working as well, you're not going to have them. And so that is, of course, what's going on in exons, because that's what's coding for your protein. Purifying selection in introns, the fact that you don't have purifying selection in introns, and you have much more diversity between two otherwise equally related organisms in your introns, means that there's not selection there, and it probably means that that intron sequence is not that important. Because if you think about conserved sequences, sequences you find in lots of different organisms, they're going to be in places where it's probably important for function. That there's no selection, okay, that's clearly not the case. Molecular clock, the key here for the molecular clock is that the molecular clock is going to be different relative to introns and exons. Because if you just think about introns where there's no selection, you'll have a certain amount of sequence change over a certain period of time. Whereas if it's under selection, then you're going to have different amounts of selection going on with time, unless you count what? What do you have to do? So if you look at synonymous substitutions, so if you look at synonymous substitutions, which are where you have a nucleotide change that doesn't change the amino acid, that you can also use to calculate a molecular clock that should be exactly the same as what you're seeing in your introns. Okay, with that, I think we've covered most of what we talked about last time, at least to some extent. We'll talk more about human chromatin, or the compaction of your chromosomal DNA in human, most eukaryotic chromosomes more next time. Exon and gene duplication, again, this is mostly through crossing over transcription units. Didn't have a question about this, but basically the idea with a transcription unit is that in most of the, as my yeast biologist friends like to tell me, larger eukaryotes, has to do with alternative splicing. So lots of different splicing that happens. Probably almost 80 to 90% of human genes have alternative splicing that takes place in them. And that, to some extent, is also reflected in the gene density that you have in some of these larger eukaryotes as opposed to some of the smaller ones. Talk a little bit about some of the repeated DNA sequences, particularly those short sequence repeats, most of which have to do with errors, if you want to call them that, in replication, where your template and your replicated strand would always match up with each other. Again, we just covered molecular clock a little bit. Synteny is a concept that I wanted to mention again. The whole idea of synteny is that you have genes which are in the same order relative to each other in multiple different genomes. And that also gives you a pretty good indication that these genomes are related to each other. So if you look at the mouse versus the human genome, very often multiple genes are right next to each other. And it's going to be true in mice and true in humans. Often those sets of genes are shuffled all over a bunch of different chromosomes, but usually those genes are relatively close to each other. And then, of course, the various different model genomes, mouse being the classic, but also some very interesting things that nobody ever would have thought of, like the fugu fish genome. Why would the fugu fish genome be a nice model genome? Well, it's because it's missing most of the extra DNA, which is in most other vertebrate genomes. So it's about 1/10 of the size of our genome, but has almost all of the same coding material in it. So today we'll finish up with protein families that we didn't get to at the end of lecture last time. Most of the time we're going to talk about transposons. There are really sort of two main groups, or flavors as I like to call them, of transposons. Either the DNA transposons or the retrotransposons, which go through a RNA intermediate. Two major classes of these, and particularly in our genome, depending on how you count it, the majority, or at least close to half of our genome, is made up of these retrotransposons. So they're a very important aspect of what we have in our genomes and true in many other of these larger eukaryotic genomes. Talk a little bit about some organelle genomes, and particularly that would be the mitochondrial and chloroplast genomes, and maybe a little bit about, they call it genomics in the textbook. There are whole courses on genomics. In fact, there are whole degrees on genomics. This is a little bit of bioinformatics. So we talked a little bit about gene duplication, how it actually happens, what is the result of gene duplication. Well, the result of gene duplication are what are these so-called gene families. And these are multiple genes that are similar to each other in sequence, usually present in the same organism. We'll talk about orthologs and paralogs right at the end if we get to it again today. Figure repeating that's a good idea. But these gene families are, again, probably something like 20% of the genes that we have in our genome, and obvious how evolution is really taking place there. So if you look at our current, in fact, alpha or beta globin gene, it's got 3 exons and 2 introns. That's true for all of our alpha-like globins, all of our beta-like globins. That gene was probably duplicated about 500 million years ago. That's well before the divergence of most mammals. And then in these duplications, and the whole prediction was, when you think about gene duplication, again, by this unequal crossing over, homologous recombination, the idea there is that you have now 2 copies of the gene. You can play with one of them and see what happens evolutionarily. And you would expect, since most mutations are actually negative, that you'd have some genes that don't work after you've mussed around with them. And that's true for all these so-called pseudogenes. These are sequences which should never function, and people have actually now done the experiments. They've tried to express these genes, look and see if they actually make a protein that works. No, they don't. And so it's exactly what you expect with this theory of making lots of copies, and then having an extra copy that evolution can basically play around with. So if you look at this, evolution is a slightly different way than the way it's outlined in our textbook. You had an ancestral globin gene, and then it ended up with this 3-exon 2-intron structure. Then there was, at some point, a gene duplication that took place. And again, this is multiple hundreds of millions of years ago. This set of genes became the beta globins. This set of genes became the alpha globins. Then, in each of these genes, they also had gene duplications, again, through unequal crossing over and recombination, whereby you end up with multiple different genes. In here, in this case, the beta globin, which is a major portion of the globin genes that we have in our red blood cells, alpha globins down here at the bottom. So these are pretty classic, but it turns out that with placental mammals, particularly if you think about the fetus, it's going to be under very different oxygen conditions than us out breathing atmosphere. And so it's actually advantageous for these placental mammals to have globins that have now different affinities for oxygen. And so it turns out that we've got a number of these fetal globins, some embryonic globin genes, etc. And so probably that was advantageous, selected for along this progression of making placental mammals. And of course, in both of these families, beta globin family at the top, alpha globin family at the bottom, you have a number of pseudogenes, and these are presumably, again, gene duplications. There's been mutations through whatever mechanism that are now no longer functional, and so these are our pseudogenes. This is exactly what you'd expect. And sort of based on these kinds of phylogenetic trees and just looking at what we have at the end, we can actually start to put together, with a molecular clock, an idea of when these kinds of gene duplications actually happened. So if you look at about 200 million years ago, that's when a lot of the fetal globin genes apparently were generated, and that's just about when placental mammals were really taking off. Whereas the actual original globin gene was probably well before that. And of course, reptiles also have to deal with picking up oxygen and moving it to various different cells, multicellularity. And so this way we can get pretty good idea, and if anything, we can even get an idea of physiology of some of these ancient organisms just by now looking at the gene sequences of what we have now. And one of the, I think, really fascinating aspects of molecular biology is we can actually now go back and make some of these ancestral genes and actually see what they look like and see how they actually function. And so you can get an idea of the physiology of organisms that were around hundreds of millions of years ago by recreating these protein sequences and then looking at how they function. I think is a really fascinating aspect of looking at biology. So any questions about the gene family stuff before we move on and talk about transposons? Okay, so transposons. And this really should be an homage to Barbara McClintock, probably one of the most amazing scientists ever, male or female, in terms of understanding what's going on with our genomes, and in fact, pretty much any larger genome that's out there that probably hasn't been selected for being really small. And basically what Barbara McClintock did was, just by doing really classical genetics, taking corn in her case and looking at normal segregation, positive selection, negative selection, dominant phenotypes, recessive phenotypes, she saw some really weird things going on. And basically, weird things like spotted kernels of corn, those that are sort of the variegated structures here, etc. And by doing, again, extremely careful genetic crosses and being really careful about keeping track of these things, she came up with some results that the only way she could describe them was by genes jumping around. And nobody believed her for about 40 years because genes didn't jump around. Well, of course, now we know that genes do jump around. And what she showed was that there was this gene, and she knew nothing about the sequences at this point. This was, again, purely genetic, purely doing crosses, that there was a gene which had apparently jumped into another gene and caused a mutation. And it turns out that that gene is a transposon. That transposon is only moved in the presence of another gene, which we now know is a transposase, which allowed this gene to jump, and this gene here made a gene product which allowed this gene to jump. And so basically it was, again, through lots and lots of crosses, she identified this DS and AC. The names are not critical, so I won't go into what they are, but that were giving these jumping genes. First people thought, okay, well, jumping genes can't happen, and then, well, okay, maybe they happen in corn, but not in other more interesting organisms like us. And again, of course, now we know with the human genome sequence that maybe up to about 50% of our genome is these jumping genes. So again, in that period of time, most of the first papers were published in the 1930s. So, quick do math, about 80 years ago now. In the meantime, we now know that there are really sort of three major different groups of transposons. The so-called DNA-only transposons, and we'll see why that is when we take a look at this in more detail. The DNA-only transposons, there are a couple of ways that you can identify these. Probably the best way is these inverted repeat sequences that you find at either end of the transposon itself, and then it has a transposase gene which is encoded right in the middle. This is probably the most common transposon, certainly in bacteria and in some animals, things like Drosophila. And in fact, it's the same kind of thing that Barbara McClintock was studying in maize in corn. On the other hand, in the human genome, and particularly mammalian genomes, you have many more of these retrotransposons, which you can tell they're different just by looking at the sequence of this transposon, because they have directly repeated sequences at either end, and they encode different genes in the middle here, particularly reverse transcriptases and integrases. And in the textbook they say, oh, well, it looks like viruses were derived from these. I, of course, think about it the other way around. I think these were all derived from viruses. And this process, again, it's a chicken and egg question. We're never really going to know. Non-retroviral retrotransposons. This is actually a really kind of interesting story, and it turns out that these are the most common of the transposons that we find in our genome. These are transposons that you actually can't recognize by these repeated sequences at either end. They just look like they have a gene-like structure to them. And we'll see how those work in just a second. So let's zoom in, starting out with these DNA-only transposons here at the top. So if you look at a classic transposon, you've got end sequences. And in this case, again, this is inverted repeats like you find in DNA-only transposons with a transposase in the middle. And the key to this and understanding how these transposons work is really these end sequences. There's two different things here. We've got the binding sites for the transposase, and this is this inverted repeat sequence right here. And then right next to them, a recognition site, or particularly this is what is present in the genome, was actually present before, and is now duplicated after you put in your transposon. So let's look at how that happes. Here we have our transposon with the transposase bound to its binding sites, and it's got a couple of 5' ends to this piece of DNA, which is just cut out of a genome from one place. Then it finds a place to bind to and cuts this DNA, ligates itself in, but in that process, it gives you gaps because it doesn't bind at just one position right in the middle of DNA. It binds and cuts at offset positions. You can see that where these two arrows are right here. So what that means is you have these gapped sequences right here with 3' OHs and templates, 3' H's and templates. I give you one guess what binds to those and does something. It would be a DNA polymerase, because 3' OH and template. So that's how it works. Here, this image is with these little balls here relative to DNA. Of course, the proteins are much bigger. Harder to see this structure, but this is basically what one of these transposases bound to DNA looks like. DNA here in red, and the two transposase subunits here in either yellow or light blue-gray. And so this is the structure which actually is now going to come down and interact with where this jumping gene is going to jump into. Let's look at what happens there. Again, it's that transposase which is bound to the transposon, but also has endonuclease activity, so cutting in the middle of a DNA molecule. It cuts, and it cuts in an offset position. And so here what you end up with is two strands of single-stranded DNA. So at this end, A, B, C, D, E, this is just your sequence of just 5 genes, nucleotides, whatever it is. This is usually a 5 to about 15 in the maximum case of any of these transposases. And the only reason that you have this gap sequence here is, again, because the transposase cuts at offset positions. And so this is A, B, C, D, E, and this should be the complement of A, B, C, D, E over here. Again, cellular polymerases, 3' OH template, replicates, you get this red sequence. Same thing over here. And that gives you now a directly repeated sequence. And what do I mean by directly repeated sequence? I mean that this is the same sequence on the same strand. So here, A, B, C, D, E, reading from 5' to 3'. Here, A, B, C, D, E, reading from 5' to 3'. So these short direct repeat sequences, again, 5 to 13 nucleotides at either end of your transposon, is an indication that you've had this insertion event that's taken place. We'll look at direct and inverted repeats a little bit more in a couple of slides. So this is the classic case of a DNA transposon where you have your DNA that you start with. This gets cut out by your transposase. It goes, finds a new piece of DNA, and inserts that DNA into that new position. Again, this is true for DNA transposons, very common in bacteria. Turns out that most of our transposons that we're carrying around with us go through a slightly different mechanism, and that's called the copy and paste mechanism opposed to cut and paste. And copy and paste basically just means that the transposon that you started with stays there, and you're making a new copy which goes and jumps somewhere else. This does happen in DNA transposons, much more common in the retrotransposons, and the reason for that is that this DNA gets copied into RNA, and it's that RNA which gets copied back into DNA. This is the reverse transcriptase, where this now can jump into a new place. And so in the case of cut and paste, you went from one transposon to one transposon, no difference. These retrotransposons, you've gone from one now to what you started with plus a new one. And so this is expanding the number of transposons, so it's actually not terribly surprising that if you look at our genome, we've got many more of these retrotransposons than we have of these DNA transposons, because these guys expand. Anytime you have transposition, you end up amplifying the numbers that you actually have of these genomes. So let's look again at these DNA transposons. I just talked about the direct repeat. This is that black sequence which you generate when you cut and paste and put this into the genome. Inverted repeat sequences at either end of the genome of your actual transposon. This is where those proteins are binding to. We'll talk a lot more about proteins binding to inverted repeats when we talk about protein-DNA interactions. But the idea is here that you've got two subunits of your transposase. You saw the yellow and the gray, which are identical in sequence, but they're in an inverse orientation. And so in inverse orientation, this guy is going to bind DNA that's going in that direction. This guy is going to bind DNA that's going in the opposite direction. So that's how you get your inverted repeat sequence. And then your protein coding region here in the middle almost always is encoding your transposase. This inverted versus direct repeats. Again, I mentioned direct repeats. We'll start down here at the bottom. Same sequence from 5' to 3' on the same strand. So GAGC here, GAGC, again, from 5' to 3'. If you look on the opposite strand, this is not the same sequence. It's from 5' to 3', it's GCTC. It's a direct repeat on the opposite strand, but it's not the same as the one you started with here. On the other hand, an inverted repeat is literally taking this sequence and flipping it on its head. So now we've got GAGC on this strand, and then on the opposite strand, you've got GAGC. So again, always 5' to 3'. Whenever you read these sequences, that's because that's how we're polymerizing. So inverted is same sequence on opposite strands. Direct repeat is the same sequence on the same strand. Make sense? No? Yes? Whatever. It's worthwhile drawing these out. The other thing that I find, and what I don't like about this particular slide, is that these nucleotides are drawn all right side up. I usually actually like to draw them upside down. Makes it, I think, a little bit easier to follow what's going on here. So if you want to know more about direct and inverted repeats, come to my office hours and we can do direct and inverted repeats until, cows come home or midterm time or whatever it is. So hopefully this is not too much beating the dead horse. Transposon, here we have each of the two transposases. Usually, again, they're the same sequence, but they're in opposite orientations. They're going to bind to inverted repeat sequences, which are these little red sequences right here. This then will cut itself out. In the case of a DNA transposon, this gets rejoined by the DNA repair machinery, goes and finds a place to bind and integrate into. Again, the cut sites for these transposases are always offset relative to each other. Because they're offset, you end up with these direct repeats right next to your transposon. So in the case of one of these DNA transposons, if you're just looking at a genome sequence, what you have is direct repeat, inverted repeat, inverted repeat, direct repeat. So it's an easy way to see if you've got a DNA transposon that's jumped into your genome. On the other hand, the retrotransposons, and particularly these are the retrotransposons that replicate in a viral-like mechanism, these just have direct repeats. So they've got a direct repeat that's come from your original genome sequence, that's that black sequence, but then this retrotransposon also has a direct repeat, and that tells you almost immediately that it's not replicating the same way as these DNA-only transposons. And the reason for that is because, at least I think, they're all due to viruses and how viruses are actually replicating here. Again, the textbook says that viruses were derived from these guys. Again, I don't believe it, and we can talk more about that in office hours later on today. So direct repeat, protein coding region, direct repeat, direct repeat. So quick overview of retrovirus replication. Reminder, you can all sign up for virology today, or at least I think all the seniors can sign up for virology today. Should be, I think, a lot of fun, but I'm also kind of biased. I know a couple of you have already had my virology course, so you know how much fun it is. And you can tell all of your friends and neighbors how wonderful retrovirus replication is. So retroviruses have these RNA genomes. This comes inside the cell. That RNA gets copied into DNA by the reverse transcriptase. That reverse transcriptase comes with the virus, so it actually is part of the virus particle that comes inside the cell. That reverse transcriptase not only is an RNA-dependent DNA polymerase, but it's also a DNA-dependent DNA polymerase. So it has both functions, making first your complementary DNA, which you've already heard about as a really useful tool for looking at messenger RNAs, but also making this double-stranded DNA. This double-stranded DNA then gets integrated into the host genome. That's a viral integrase which does that. Then this integrated DNA gets transcribed. Each of these RNAs can both encode viral proteins, but also the viral RNA, which then gets put together into a virus particle and is then released, goes off, and infects yet another cell. Of course, the main important enzyme for this is the reverse transcriptase. This is a classic DNA polymerase. It takes 3' OHs and extends them. The only big difference is it can use RNA as a template as well as DNA. So it can have both of those. If you look at the structure, of course, all of you recognize this is a DNA polymerase-like structure, right? Well, maybe not exactly. But yes, it's a DNA polymerase-like structure. And again, has a 3' OH, which then can be extended. Does not have a proofreading activity. So one of the things you've probably heard a lot about, HIV, lots of mutations. The reason for that is that this guy doesn't have any proofreading activity. It's one of the reasons for that. So we look at this in a little bit more detail. I won't be testing you too much on this term. Take my class next term, we'll have a lot more about it. But one of the things you should be asking yourself is if this is a classic DNA polymerase, it needs a 3' OH, right? So you have to have that 3' OH coming from somewhere. Well, where's that 3' OH coming from? There's no DNA primase that you're talking about here. So it turns out that these retroviruses have evolved to use cellular RNAs as primers. So they use these tRNAs, and it's in fact a specific tRNA inside the cell as a primer, which provides a 3' OH. That then extends, interestingly enough, just through one short piece of the end of your genome and through a very convoluted process that, again, we'll talk a lot more about in virology next term. You end up with copies of this end, what you started with in your RNA, at both ends of your DNA down here at the bottom. And these are now those direct repeat sequences that you find in all of these virus-like retrotransposons. So it's just the replication process of the virus and also all of these LTR-like retrotransposons, which will generate this sequence at the bottom. And it's all because reverse transcriptase is a normal DNA polymerase. It can't prime by itself. It needs to have a primer of some kind, and that primer, in this case, is the tRNA. So that's what's happening with our DNA transposons, our viral-like retrotransposons, but confusingly enough, most of the retrotransposons in our genome don't look like these viral transposons. They actually look kind of different. And that difference is they've still got these direct repeat sequences at either end of the transposon, and that's because all transposases cut slightly offset at either end, which you got to fill in. 3' OH gets filled into the end. But now instead of having direct repeats at either end, like you have with these retroviral-like retrotransposons, now you have these AT-rich regions, and particularly this end right here, which looks like a normal poly-A tail that's gotten added to regular messenger RNA with then a poly-T in the opposite direction because that's been copied back into DNA. These protein coding regions that you have in the middle here, they are clearly a reverse transcriptase, so looks a lot like the reverse transcriptase that you have in these viral LTR. Oh, LTR, by the way, is long terminal repeat. Sorry, I should have mentioned that before. So long terminal repeat is that piece which gets made from the tRNA primer. And it's a direct repeat sequence, so you end up with 2 copies of that when you're integrated into the genome. So these guys don't have those LTRs, but they do have a reverse transcriptase and also an endonuclease, which is present in their genome. This particular cartoon is of the long interspersed, and in fact there should be nuclear element right in here. The line element, this is the second most common transposon that we have in our genome. So very common. We don't actually completely know how these guys replicate, but we can guess, and we've got more and more indications that this is what's actually going on here. You end up with, again, one of these transposons inserted into your genome. That gets transcribed into a normal messenger RNA with a normal poly-A tail on it. there is an endonuclease, and again, this is the product of that ORF2 protein, which will make a cut in the genome where you're going to be inserting this. And then the reverse transcriptase uses the messenger RNA as a template to make one copy. That reverse transcriptase will then also make the second DNA copy, and then this piece gets integrated into your cellular DNA, leaving, as always, these gap sequences which get filled in by the cellular polymerases. So I mentioned that line elements are the second most common. The most common are the so-called sign elements, or short interspersed nuclear repeats. Turns out that they're basically identical to these line elements, only they don't have the reverse transcriptase and the endonuclease in them. And that, in fact, makes reasonable sense because as soon as you've made this protein, it's a messenger RNA, it's got to get excerpted from the nucleus, gets translated into protein, that protein comes back into the nucleus, that protein is just going to be recognizing DNA sequences. It could care less whether that DNA sequence encodes a protein or doesn't encode a protein. So all you really need is the end sequences of any of these elements to get that to move around. Because as long as you've got some of the transposase protein, some of the endonuclease protein, it'll bind to the ends and insert them. And that's, in fact, what was going on in Barbara McClintock's case. She had DS, which, as it turns out, was just a transposon that didn't have a transposase gene in it, and AC, which was the transposase gene, which could actually then move through the cell after it had been translated and act on this, also called a mini transposon. And so that, it turns out, is also extremely useful for using transposons in molecular biology. So when I'll talk a little bit more about that, transposons are wonderful tools. They're just natural mutagens by themselves, and so you can induce transposons to move around inside the cell. There are lots of different ways to do that. We won't get into it right now. But if you're going to induce transposons to move, you will generate mutants because they're insertions. And if they happen to insert in the middle of a coding gene or some kind of regulatory sequence, it's going to generate mutants. Once you've done that, you can determine really easily where that transposon is inserted, either by the transposon sequencing that we talked about a couple lectures ago, where you have a restriction endonuclease site in your transposon that you then cut. You then cut also your genome, which is going to be near that, religate these ends together because they've got cohesive sticky ends. You can sequence that because you know what the sequence is from your transposon. And so you just either amplify with sequences in your transposon or sequence from your transposon. These so-called non-autonomous transposons, when you say non-autonomous, that means that they are not encoding the transposase or the reverse transcriptase endonuclease, so something like a sign element, or again, those DS elements that Barbara McClintock was working with. These are really nice because once they've jumped, there's no transposase around anymore. They're stuck there. They're not going to move around. And so you can generate very stable mutants this way. And what we've done, we being the molecular biology community, many of them have these selective markers. So let's take a quick look at some of these. Your normal transposon is just like this. And this is their DNA transposons. You can do this with retrotransposons. It's actually a lot harder to do that. So most of the time we're using DNA-only transposons here. So normally you have your transposase with these inverted repeat sequences at either end. You can just replace that transposase gene with a drug resistance gene. And now this is a non-autonomous element. If you put in transposase, either expressed from another gene, or in many cases you actually do this in a test tube where you put in some transposase protein, and then these transposons will jump around. Quick reminder, those of you still looking for lab classes next term, we're actually going to be doing this in the recombinant DNA lab, is literally putting in transposon mutants in one of my viral genomes. You can also have much more complicated versions of these transposons that have reporter genes and drug resistance genes. So this particular reporter gene, like we talked about last time, could be something like a beta-galactosidase, a green fluorescent protein, in some cases luciferase. And basically what this reporter does is when the transposon jumps around, sometimes it's going to land in a part of your genome which is being actively transcribed. And then you've got a reporter gene that says, hey, this part of the genome is being actively transcribed. Now I want to sequence this part of the genome and say, what is that gene which is being actively transcribed here? And then in some cases, there'll be a promoter that you put into your transposon, which now will express a gene right next to it, even when that gene is not supposed to be being expressed. And this is a way that you can turn on genes in particular cells under much less normal conditions. So it's a nice way of figuring out what happens when you overexpress a particular gene. So these are just a couple of neat tools that you can do using transposition and transposons. Just one other example of that is this so-called transposon library. This particular case, the transposon is being introduced with a bacteriophage that has transposase and the transposon on it. As long as you've got a gene, usually an antibiotic resistance gene, again, in this artificial transposon, you get insertions all throughout your genome. And using PCR, you can amplify all of these transposons. You get a particular piece of DNA which you can now sequence. And usually this is going to be using one of these high throughput DNA sequencing mechanisms. So pyrosequencing, Illumina sequencing, etc. Basically what you're doing here is you grow the cells under one condition with your transposon library. You grow cells under another condition with your same transposon library. And basically what you're looking for is where you don't have any copies of this particular mutant. So mutant, again, do the transposon insertion. And so here what you're looking for is the lack of this one particular product here. That lack of that one particular product means it's probably important for growth under these conditions. And so it's a quick way of identifying where you've made a particular mutant. We also talked a little bit about transposon sequencing before, where you can just find out where your particular transposon is inserted into a piece of genomic DNA. But also, just the fact that you know the sequence at the ends of your transposon means that just by putting a bunch of transposons into a large piece of DNA, this is identified as this large circle down here, you can now sequence using primers that are in your transposon through the rest of that piece of DNA. So instead of doing what's called primer walking here, one of the big problems with sequencing big pieces of DNA, and I talked about shotgun sequencing a little bit before, and I talked about it in office hours too, is that usually when you're sequencing, DNA polymerases have this problem, just like reverse transcriptase. They have to have primers. So you have to put in a primer of known sequence when you're actually trying to sequence something. Well, if you're trying to sequence through a really big piece of DNA, and you don't know what the sequence is of a lot of that DNA, you have to generate the sequence data, then design the primer based on that sequence data to sequence the next piece of DNA. And so that's what's shown up here on the top, what's called so-called primer walking. So you do this sequence, then you have the data for this sequence, you design this primer that's complementary to that sequence, and so on and so forth. And you walk your way through the genome, walking your way through a big piece of DNA. On the other hand, if you have a transposon library where lots of transposons have jumped into this piece of DNA, you know the sequence of your transposon. And so you can just make a primer which is complementary to the sequences at either end of your transposon and sequence from there. And so as long as you have enough transposons that have integrated into your large piece of DNA, then you can generate all the sequences of that big piece of DNA all at the same time. So instead of having to know what the sequence is of any of these individual pieces. So there are questions about transposons before we move on and talk a little bit about mitochondria and chloroplasts. Yes?
So as far as like where the transposon inserts itself,
Mm-hmm.
talking about like putting it in specific places. So how would you determine that?
Okay, so
Determine that itself.
okay, maybe I misspoke. So the question here is, since I'm also recording this, is how does the transposon know where to jump into, or do you know where the transposon is?
Yeah, like does it have like specific?
Right, basically specific sequences that the transposon jumps into. In theory, transposons jump into genomes randomly, and so you only know where that transposon sequence is after you've done another experiment. That would either be sequencing, and then you can identify a sequence next to where that transposon is. And so I may have misspoke earlier, but the transposons should be jumping in randomly into your DNA because they should be nonspecific. There are exceptions to that, and the most immediate one is some of you may have noticed when we talked about line element and sign elements. They've all got poly A tails on them, and in order to get your replication of that opposite strand, you have to have a run of T's to be able to start from. So then in that case, you clearly have things that are jumping into slightly, not completely random places. Other questions on transposons, direct repeats? Yeah?
Doesn't the transposase it has to bind to a very specific, like, complementary sequence on it before it inserts?
Yeah, so the question here so isn't the transposase binding specifically? And yes, the transposase is binding specifically, but it's binding specifically to the transposon itself. Once it's bound specifically to the transposon, when it goes and cuts somewhere else in the genome, that's a random position that it binds to. So the binding to the transposon is specific. That's where you have your inverted repeat sequences. It's once you've bound, you've got your, I avoided talking about this because I don't like the terminology, but we had that figure where we had the two transposase subunits bound to the DNA. People call that the transpososome. Again, horrible name, but that's what people use. But that transpososome then is very nonspecific. So at that point, the transposase is bound to the ends of the transposon. It can basically go anywhere and cut, offset, and then incorporate right there. Again, with the exception of things like those line and sign elements where it has to at least be an area where you've got a template that you need to combine to. Yeah?
This really doesn't have anything to do with what we're talking about. The last week, right before we left class, you said that there was a conserved element in DNA that was not an exon.
Yes.
And you told us to think about it.
Yeah.
And now I'm thinking about it. Have you expounded on that?
So, okay, again, just so that our audience online can hear the question. What about those things I told you to think about last time, the conserved sequences between multiple different organisms that aren't in exons? And so there are a number of different things that are going on there, but probably the most obvious of those are small non-coding RNAs. And so they're not in exons, but they're really important regulatory RNAs.
So they're not transposons?
They're not transposons. They're actually usually shorter than the transposons, those particular sequences I was talking about. They could be transposons, but most of these transposons, at least the autonomous transposons, have a transposase gene or a reverse transcriptase gene or an endonuclease gene in them. Those you can see. Those are coding sequences.
I thought they might be signs.
Yeah, they could be, some of those could be the signs. It turns out that there are different enough sequences from different organisms, strangely enough, that's often not the case. So usually these are the small RNAs, which you're finding there. And we'll talk much more about those small RNAs when we get towards the end of the course. So not completely off topic, but close. So last 10 minutes or so, I want to talk a little bit about endosymbionts. And the critical aspect here with endosymbionts is basically mitochondria and chloroplasts are really a lot more like bacteria in terms of how they're replicating, how they're doing their jobs. And we know this mostly because of comparisons of sequence. If you start to look at the sequences of DNAs that you have in mitochondria sequences of DNA that you have in chloroplasts. These are much more similar to bacterial DNA than they are to the eukaryotic DNA that you have in the nucleus of these cells. So what seems to have happened, probably literally billions of years ago, there was an ancestral cell which then picked up a bacterium, probably through some kind of endocytosis. That bacterium then became a mitochondria. In other cases, and this has actually probably happened later, there was a photosynthetic bacteria that got picked up by this ancestral cell. I'm not calling it a eukaryotic or archaeal cell. People argue about that a lot. So an ancestral cell that then picked up one of these photosynthetic microorganisms, and basically this now developed into all known plants, at least photosynthetic plants, due to this picking up of the chloroplast, and here picking up of the mitochondria. If you look at mitochondria with just DNA binding stains, here's the nucleus, and here is all of the mitochondrial DNA. And so basically what this is telling you, if you think about DNA in a eukaryotic cell, a regular eukaryotic cell, it's not just the DNA that you have in the nucleus. You also have DNA in mitochondria and chloroplasts. And so when people talk about the DNA complement of a eukaryotic cell, it's not just the nuclear DNA. And people will also really specifically say nuclear DNA versus the non-nuclear DNA. If you think about mitochondria, mitochondria are really kind of interesting because that DNA that you have in them, and the same thing is true for chloroplasts as well, is that all of that DNA is inherited maternally. And so it's not a nice Mendelian inheritance pattern that takes place. And the reason for that is that certainly if you think about most animal reproduction, sperms are wimps because they really just pretty much have the genetic machinery. They don't have everything else that they need to make an organism. And so all of the other things you need to make the organism come from the egg. And so that is where all of your mitochondria are coming from. If you do this in yeast, which of course is not male and female and has different ways of reproducing, A's and alphas, which actually is a cell fusion event that takes place. Here you have particular kinds of mitochondria, and these are actually defective mitochondria here. If a cell with defective mitochondria mates with one with normal mitochondria, it turns out that this is perfectly happy because it just passes all of these things down. However, if you have cell division that takes place with one of these cells that has some unhappy mitochondria, eventually these unhappy mitochondria are going to end up being the only mitochondria which are around. And so these cells are really quite unhappy. If you look at the genes that are encoded by the mitochondria, it's actually not surprising that most of them are involved in energy metabolism, particularly ATP manufacture. So this particular figure here, everything which is in pink is a clearly mitochondrial gene. Everything that's in blue is an interesting kind of protein because it's expressed in the mitochondria, but it's a nuclearly encoded gene. And so that tells you that this symbiosis, when you had that incorporation of that cell, again, particularly for the mitochondria, probably billions of years ago, a lot of the genes that used to be in the mitochondria are actually now in the nuclear genome. Now, how that process took place, how you took a gene from a bacterium and transferred it and put it into a eukaryotic nucleus is a really fascinating question. Nobody really knows. You want to get a Nobel Prize, figure it out. But a lot of these things, and this is true for animal cells. Curiously enough, other cells actually have different kinds of mitochondrial genomes. And we have one of our faculty members here in the biology department, Dr. Susan Masta, who's actually looking at mitochondria and why different organisms have different kinds of mitochondrial genes that are associated with them. So one other aspect, we talked about different genetic codes before. Probably the best known different genetic codes are those that you find in mitochondrial genes. And so if you look at the standard genetic code, UGA encodes a stop codon, but in mammals, flies, Neurospora, some other yeasts, it all encodes tryptophan. And so this is the classic recoding that's taken place there. Some other codons here, AGA, stop codons, etc. I don't expect you to know this whole table, but just that coding in mitochondria is different. And the reason for that is that there are only a few tRNA genes that are encoded in the mitochondria. And since you've got fewer tRNA genes, then you're going to have fewer possibilities for wobble, etc., that's happening in terms of your genetic code. So it seems that the mitochondria can get away with having fewer tRNAs, whereas the host cell can't. And probably it's just because, again, of this relatively small number of genes that you have present in your mitochondria and those that are actually being expressed. And it turns out from organism to organism, these are slightly different relative to each other. Wanted to show you one example here, and again, this is from the textbook, about some problems with mitochondria, which can be really major issues. So here, this is just a case for mice. These are mice that have a mitochondrial DNA polymerase that lacks a proofreading activity. The wild-type mouse is perfectly happy. A heterozygous mouse is relatively happy, but homozygous mice are really unhappy. And the reason that I wanted to mention this is a number of you may have heard in the media about a researcher at OHSU who's actually now been able to take mitochondria from one cell and transfer them into another cell. And he's done this now in primate cells and also in human cells. And this brings up the really interesting possibility of a human having potentially 3 parents, because they can have a mitochondrial parent and then nuclear parents. So you'd end up with 3 different parentage. And there are some nasty mitochondrial diseases. This particular one turns out not to be a problem in humans, but there are a number of mitochondrial diseases. So if you can get mitochondria from somewhere else, well, that's genetic material from someone else. And so how do you think about that in terms of an ethical issue? And if you want to ask about that next time in lecture, you can think about that until then as well. So we'll see you all on Wednesday.